it works.
Okay.
For example, here, this is one of my protein that is called TIDP1, human
Mm.
protein, tyrosyl DNA phosphodiesterase 1,
Okay.
which has around 608 amino acids, as you can see here.
Yeah.
And then it is the gene protein of Homo sapiens human. Okay.
Yeah.
And then you go for all the information. The function of this particular protein is mentioned here.
Mm.
Then you go for kinetics. Okay, Km value is 0.08. And then you calculate, find many more things by following the links. Then it is saying active site. The sequence classification is something like active site, binding site, active site, binding site, normal sites, that is interaction with DNA, active site, nucleophile, substrate, proton donor acceptor, and then substrate binding sites. For these, the predictions, it is known from these two publications. If you will click here, you will get the publications who have published the.
Brother, just a second. My roommate is just knocking. Just a second. Majid bhai, সাউন্ড আসতেছে ইয়ার আসিফ ভাই যে রুমে ছিল ওইখান থেকে। এটা কি নেট আমি ইয়ে হয়ে যাবে sorry bro, like in downstairs one of the like like what is called the detector, smoke detector is running on, but no one is there. It's sometimes
Okay.
it does when battery is
Okay.
running out. Yeah, sorry.
So now you got the idea. If we want to see these two publications where they have shown at position number 263, there is active site for what? A nucleophile.
Okay.
Similarly, 265 is binding site for a substrate. And many more things you can see by clicking the link. Now the detailed description of this particular protein, that which sequence could be doing which function, for example, all molecular functions, then DNA binding activity is possible here, then hydrolase activity is possible, mitotic cell cycle is controlled, DNA repair is also where it plays the role. In the similar way, you can see the functions that nuclear component, like cellular component, is also available, nucleus, nucleoplasm, then plasma membrane, where it is present, organelle control, and other cellular components are also where it remains present.
So it's illustrating like which part of the
This is basically called, we will be coming about this point very soon when we
okay.
will go talking about functional annotation.
Okay.
But I'm just trying to show many informations you are getting more than anywhere on uniprot.org.
Okay.
Now, this is about subcellular location, that where is the location of this particular protein, which you can see here. Okay.
Yeah.
So this is for this. Then you click disease and variations. What are the diseases known to be associated with this protein? Okay. And then for post-translational modification, where are the phosphorylation sites or some other sites which is already coded here.
Mm.
Then you go for expression of this particular protein. Then the expression is also possible, but in which term you have to select here, where it is expressed in the cellular location or subcellular location. Then you can go for disease specific things. Everything is available here.
Okay.
Now you can see the structure of this particular protein. The structure is here. So you don't need to go to PDB also. You can get some information here for most of the proteins.
Okay.
In case if you don't get the link, then you have to go to PDB or
PDB.
go for prediction.
Okay.
Is it clear now?
Yeah.
Yes. Now I was saying that there is something called AlphaFold And here is the link. If you want AlphaFold 3D structure, you click here, you will get the AlphaFold 3D structure. Is it good?
Yeah.
Yes. Now the protein classes family. What is the family of this particular protein? What are the domains here? Everything you are getting the information here. Okay.
Yeah, bro.
So there are many more things you get. Now it is the term of for getting sequences. This is the primary structure of the protein.
Yeah.
If you will click here, download, it will give you this something like this. And you just need to copy this link, not link, the sequence.
Sequence. Yeah.
Just leave this greater than symbol line,
Hmm.
copy all the sequences. And if you want to predict the 3D structure, you can use any software like a Swiss model
Swiss
or
model.
some AlphaFold or anything. You
Hmm.
will get it immediately.
Hmm.
Okay. Start modeling, then quickly paste here, and then something like XX, and then go for build model. Click here, build model, not search for template, build model.
Hmm.
You will get at certain time 3D structure, and you can download the structure. So this is like the details of some of the database, and this is the most common database. That is why I have shown you.
Okay.
Now, computationally mapped potential isoform sequences, and there are 13 sequences which are supposed to be the isoform of this particular protein
Okay.
or similar protein. So there are large number of information which you can get from here. I hope it is very clear now.
Yeah, of course.
Yes. So this was one of the database.
Database.
Okay. Now this is the introduction about the database. I want to make you more informative in the sense of pathways. Okay.
Okay.
So if you want to go for pathway information, then what you need to do is basically, for example, I have clicked on 4390. This is hippo signaling pathway.
Okay.
But you have to go something like in the very basic instead of following my steps. This is the home page
Okay.
of KEG database. Okay.
Yeah.
Here you just need to write any pathway name,
Hmm.
pathway name. For example, I'm writing wind signaling.
Hmm.
Okay.
Hmm.
And then organism you can select here, or you can, even if you don't select, just ignore it and go for click. They will give you all the pathway name. You just select which are the human signaling,
Okay.
like, and here 04310. And what it says is basically wind signaling pathway. Okay.
Hmm.
You
Hmm.
can try to check if it is human or not.
Hmm.
Then I clicked here, what it says, wind signaling pathway. And then we can find if it is Homo sapiens. It is not mentioning anything, but this is Homo sapiens. Okay.
Yeah.
So you can also check there directly. You can put the Homo sapiens name and then you can commit here. Okay.
Yeah.
For Homo sapiens, you just need to write HSP or something. Then it will give you. So here you just click on this link, map 04310.
Hmm.
It will give you this kind of pathway.
Okay.
This is the overall pathway. in terms of a plot kind of things. You can see most of the genes named here, most of the genes.
Mm.
Okay.
Mm.
And then you can also, basically, this is a plasma membrane. You can also understand